Hi, everyone. My name is Euan Joly-Smith, and I'm a PhD candidate in the Department of Physics at the University of Toronto. And so here I added the word dynamics to the title because I'd like to share with you one of our results involving dynamics. In particular, here's a population of genetically identical cells. They're all expressing the same fluorescent protein. And as we can see, there's significant cell-to-cell variability. This is because gene expression is a stochastic process. mRNA and protein numbers exist in relatively small abundances such that the random nature of the chemical reactions underlying gene expression becomes significant. So how can we exploit this variability to infer dynamics in gene regulation? So this variability is often called noise, but this is a little misleading because not all variability is noise. So for instance, if we take the blue here to be the protein concentration or the fluorescence and red to be the transcription rate, maybe the transcription rate is varying stochastically and so is the protein concentration. Or maybe there's deterministic variability. For instance, maybe the transcription rate is oscillating and what we're seeing is simply unsynchronized cells. Or it could be more complicated than that. For instance, maybe there's some kind of feedback control mechanism where the transcription rate has to vary significantly in order to compensate for fluctuations in the protein concentration in order to keep it at a fixed level. So from this variability, can we distinguish between these kinds of dynamics? And FYI, in this picture is the repressor. So this picture would fall more closely into this category. So this question that I posed is difficult because high throughput methods give static population snapshots. What I mean by that, techniques like single cell RNA-seq or flow cytometry, they measure the abundance of mRNA or proteins at one moment in time for each cell. So after you analyze your data, you end up with a distribution. And the measure that's most often used to quantify this variability is called the CV, which is just the standard deviation divided by the mean. So from these CVs, from these static snapshots, can we infer dynamics in the transcription rate? To answer that question, we need to ask, how do different types of dynamics affect downstream variability? And so one type of dynamics that affects variability is delays. To see that, we can look at something that we're all familiar with at this point, namely delays in the reports of COVID-19 case numbers. Because COVID-19 reports take time to be analyzed, there's a delay between the observed number of cases and the actual number of cases. Now, if this delay was 2 weeks, let's say, for each individual, then the observed trace would just be a translation of the actual time trace. And so the variability of the two traces would be the same. But in reality, the delays are distributed. For some people, it could take a few days. For others, it could take a couple of weeks. And so what happens is that the height of this curve here flattens. The variability of the observed curve is different than the actual curve. So here we can see the variability as being the height of the peak. So this is called time averaging. And the exact same thing happens with fluorescent proteins because fluorescent proteins take time to mature and fluoresce. Once a fluorescent protein is translated, it has to undergo certain conformational changes before it can fluoresce and we can detect it. And the time it takes to do this is called the maturation time, and it ranges from minutes to hours. So typically, fluorescent proteins with long maturation times are avoided in order to avoid this time averaging effect. But we wanted to see if we could somehow use this experimental nuisance to our advantage. So here is the idea. You take two genes, two fluorescent proteins, and you engineer them to have the same transcription rate. So they have the same promoter. One has a fast maturation time and one has a slow maturation time. So they're reading out the same signal, but on different time scales. Then from the population, you can measure the CVs from the static snapshots. And can we somehow use these CVs to infer dynamics in the transcription rate? The good news, this is just a dual reporter. This is the same technique that Michael Elowitz and others have used to quantify sources of variability in gene expression, intrinsic and extrinsic contributions to gene expression. The only difference is that here we require that the maturation times of the fluorescent proteins are different. So we wanted to come up with a theory, but we wanted to make as few assumptions as possible. So instead of looking at a single model, we looked at entire classes of systems. So these are classes of stochastic reaction networks that model two fluorescent proteins that are co-regulated. They have the same transcription rate. The transcription rate here can be any time-varying signal. It's unspecified, and it can depend in an arbitrary way on an unspecified cloud of time-varying components. The two assumptions we make are that the maturation step, its kinetics, is a first-order process. So the maturation times follow an exponential distribution. And the fluorescent proteins are stable such that the numbers are being reduced solely from cell division and dilution. So this is a large class of systems, so you can't solve for it. But you can analytically solve for bounds on what is possible. So for instance, if R of t is the transcription rate, any system with a stochastic transcription rate, defined as a system with a non-negative autocorrelation, has to lie in this region. So at the bottom here is the ratio of CDs, and on the y-axis is the maturation time ratio of the two fluorescent proteins. Systems that are driven by an oscillation, a deterministic oscillation, on the other hand, can lie anywhere within these orange bounds, including this region as well. But in biology, there are no true deterministic oscillations. There's always going to be a little bit of noise. So we define an oscillation as a signal with an autocorrelation that becomes negative at least once, so as to include these noisy oscillations into our definition of an oscillation. And these systems can also lie anywhere. So the idea here, if you measure this ratio of CVs, fermiostatic snapshots, and your point was to the left of this dashed black line, then you'd have to conclude that your system is driven by an oscillation, and that's without any time series data, thus turning an experimental nuisance into an analysis feature. So the theory for this constraint has been published, but in the process, the reviewers brought up a very important point, namely, would this even work in a real biological system? So as a theorist, I somehow convinced myself that the best way to answer this question was to do the experiment myself. Because if I could do it, then surely this ought to work, and surely the experimentalists would be able to do it. So I joined a lab in Montreal, and I learned how to do these experiments. And here is the experiment that we did. We used a microfluidic device called the mother machine, which has these small trenches that can hold single cells that grow and divide. The daughter cells get pushed downwards and media takes them away, but we can follow the mother cells for many divisions. So we can follow them over time. Then we took the repressor, a synthetic oscillator made up of three proteins that repress each other, and we hooked it up to two fluorescent proteins, one with a fast maturation time and one with a slow maturation time. So they're both being repressed by TEDR. They have the same promoter. And so the reason why we chose this setup is because we can use the time series data as a control to see that there are oscillations, right? But the method is high throughput enough, this microfluidic device can hold enough cells such that we can combine all of the data into a distribution and then see if the CVs from the distribution agree with what we see, right? So here's the result of the experiment. This is an example of a time trace. We have the distribution from the data, and the point lies right here. So was the referee right? Well, remember, this is an oscillation, and oscillations can lie anywhere here. So this doesn't disprove the theory, but the point doesn't lie in this region where this would be actually useful. However, note here that the period is 3 hours and the longest maturation time is 30 minutes. So the period is pretty long compared to the maturation times. And so the maturation, there's not a lot of time averaging occurring because the maturation times have enough, the fluorescent proteins have enough time to adjust to the oscillations. So what we did is we took a version of the repressor with a faster period, right? And now we can see the time averaging occurring. We can see there's a difference in the variability. The red doesn't quite catch up to the oscillations. And as we can see, the point lies right here, breaking the bound just barely. So was the referee wrong? Well, I think the bet is still on because the point is pretty close to this boundary, and ideally we'd like to get it further to the left. So the theory predicts that this point will move further to the left if we take a very short maturation time and a very long maturation time in combination. So currently, I'm redoing the same experiment, but with different pairs of fluorescent proteins to try to see if we can optimize this, this technique and move the point further into the discriminatory region. One thing that I have done so far is we've done the same experiments, but with a stochastic transcription rate. And the point does lie in this region where the theory predicts it ought to lie. So that concludes the result I want to show you on the dynamics. I just wanted to add that using this framework of classes of dual reporter systems, we've been able to derive other bounds and constraints that promise to allow us to exploit them to infer other features of gene expression or gene regulation. For instance, we derive a bound that we can use to detect feedback in gene regulation. So if you're interested in that, you can look at our paper here. I also want to add that these constraints, we've been able to show that they hold in terms of mRNA abundances instead of just fluorescent proteins. And currently, we have a set of constraints from the theory, and we're experimentally testing them using variants of the repressed later and other synthetic circuits like the experiment that I showed you so far. So with that, I'd like to thank Andreas Hilfinger, my supervisor at the University of Toronto, Jerry Wang, who was an undergrad in the group and who's now a PhD candidate at Caltech, Laurent Potvin-Trottier and his lab for inviting me and for showing me how to do these experiments. And of course, if anybody has any questions or suggestions, feel free to email me. I'd be happy to chat. Thank you very much.